Hello everyone, Assalamu alaikum. Previously we've talked about the concepts of HMM in detail, their terminologies, the Bayes theorem to predict how we calculate the probability of each site moving from one transition hidden state to the next state, or the transition probabilities and the emission probabilities using one formula. So now if we move to the example of protein profile using HMM, this is one of the major approach which is used for profiling protein sequences to identify different patterns or conserved sites. So let's take this example. There's a number of there are 5 different sequences, and the total length of each sequence is 8, and each color represent each sequence. And here you can also see that there is one insertion at this site. These dash bar represents deletions, and this gray side represents an insertion. So first, if we ignore the insertions and just focus on the probability, emission probability at each site, that what is the possibility that each site contains any of these numbers, right? So let's see if we talk about the first site. Here you can see that A is repeating at each site from 1 to 5th sequence. So the total probability out of 1 of having A at this site according to this sequence is 1, because there is no other sequence which is present at the sequence 1 at this site. So this is all these sequences contain A. If we talk about the next here, if you see here that the possibility of having C at this site is 1 plus 1, 2. So 2 out of 4 times C is present, 1 out of 4 times F is present, and 1 out of 4 times D is present. So this is the way it calculates the probability of at each site, here, 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 all these different uh, positions. So if we make an HMM diagram of each all this, so we can say that there's a probability of 1 of having A at this site into the probability of 0.25 having D at this site, 0.75 having D at this site, and so on for the rest of the sequences, right? So now, what about insertions? If we talk about insertions at this site, for example, there is a sequence like A D D A F F D F, what we talked previously in the previous slide. What if there is a probability or there is an insertion at any site? So how to calculate the probability of having F at this site between A and D, right? So for that, we just, after adding the matches at each site, for example, at from M1 match site to M2 match site 2, 3, 4, 5, 6, 7, 8, then there is also a possibility that M1 and M2 can have a probability of insertion at this site between M1 and M2. So then there can be so we will add another insertion state here, and we can say that from there's a possibility that N1 can move to I1 with an insertion. There can be probability of any probability of moving M1 to I1, and then there's any probability of moving I1 to M2, and also there's a probability of again having insertion at this site. So we add insertions on all these sites, and here we can see that from uh, I not first site to the last site, there is a possibility of insertion at each of these sequences, right? So again, then in the next slide, let's see how deletions work. So now after insertions, there is one more thing that we have shown you in the previous slide, that was deletions. So each site in a model, for example at M site here, let's say that this is a site M, so there's an equal probability of an insertion after this site, and then there's also an equal probability of deletion at that site, right? So then again, but one important thing you should keep in your mind, that we cannot directly, HMM model does not talk about jumping from one site directly to this site for insertion, and this site to this site for insertion. So these are not applicable in an HMM model. However, this, if you look logically, all these insertions which are interdependent on the previous site are also linked to this side. For instance, if we are talking about a deletion at M phase, it will moving from this deletion to the possibility of another transition probability to another deletion site and this deletion site, and then it can move to here. So the the algorithm for HMM is about the probability from one previous state to the next, not rather than from it can jump from one side to another, as we have also talked about that it is a memoryless uh, algorithm, right? So here you can see that the after insertion of each side, inserting an insertion probability at each side, now we have also included the probability of deletions at each side, and lastly, after including all the possible insertions and deletions and the emission and all the emission sites from M1 to M6. Lastly, the model also add one start site of the model, possible start site, and one end site. So yes, so from here you cannot move backwards, as you can see that from end probability this is not going back into the deletion probability, right? So these are the this you can say that this is the generalized model for profiling of protein sequences after calculating the probability at each site for any insertion, deletion, or any uh, other possibility, right? So if we calculate the hidden path through the profile, for example, here if we talk about the from first sequence A, C, D, E, this one, E, F, A, C, A, D, F. So we can see here that there is a possibility of A at this side, then C at this side, D, E, F, A, D, F at each site at M1, M2, M3, M4, M5, M6, right, and M7. So, but if you look closely here, that there is a possible insertion of AC at this site in the first sequence. Then if you look at the next here, you can see that the color actually represents the sequence. Here if you see that the green represents the first sequence, red is representing the second sequence, and then there is a, but at this side there is a possibility of deletion from A to, from M4 to D, right, D5. So because here you can see that there is a deletion at this site, and similarly if you move to the next here for the purple sequence A, then there's a deletion, then there's a deletion, then there's a E, here I'm talking about this one, E, then there's a deletion F, and then not the deletion, sorry, I just make it more clear. For example, here, right here you can see A for the purple sequence, then there's a deletion, then there's a deletion, then there's a sequence here, then there is a sequence here, then there is an insertion, and then there is a deletion, and then F, D, C, these four sequences. So if you look at here, you can see that at A side there is, after A side there's a deletion at D2, and then again there's a deletion at D3, and then move forward, right? So, and so on. And if we move to the next sequence, let's see, we have here a sequence of these five which I've shown in the previous slide. Let's see if we talk about the fifth site from match 1, M1, M2, M3, M4, and 5. So here if you see that from at this site, from each M5 position of each sequence, here there's a possibility of insertion at the first site, here there's a possibility of insertion here, and then there is a possibility, then there is a deletion at this side, so we'll not consider it because we are talking about the insertions here or the any other transitions from this side. So at this point there is a possibility of insertion of D at this side from between M5 and M6. Then there is a possibility of match, there is no insertion here, from M1 to M5 to M6, and then at the last side there is a possibility of Again, there's a possibility of insertion at this site. So if we calculate all these transitions, so we can say that there is a total number of transitions at these sites are 4, right? And in these 4, 3 out of 4 are actually the insertions, and only one is, you can say, transition from match site to the next match. So the total possibility of insertion at this site is 0.75, and the possibility of match is 0.25. So this is how you calculate, in the most simplest way, the probability of insertions or deletions or any match site at each position in a nucleotide or amino acid. So in the previous slide, we talked about the transition probabilities from one site to another, whether that can be insertion or there can be from match to match. So now if we there's one more simple concept, that is the emission probabilities. So at each site, for example, if we consider here this site 2 of M2, so from at green, red, purple, and brown and blue, all these are the different representing different sequences. So if we calculate the emission probability of, what is the emission probability of C at a site, F at a site, and then again C at a site, and then D at a site, M2, what is the probability of each? So for that, if we just look into the details, for instance, here you can see that if we look closely, that C is actually occurring 1, and then uh, twice, right? So it's occurring twice. So we can say that the possibility of the emission of C at M2 is 0.50, or you can say 50%, or 4 out of 2 times, 2 out of 4 times C is occurring at M2. And similarly, if we talk about the probability of F at a site, so here we can see that this one is F, so it's not maybe clear here. So if we calculate the probability of F, so we can see that F is occurring at this site, so the probability of F is 1 out of 4. And again, if we talk about D, uh, this one is 1 out of 4, right? So again, if we talk about D, so D is occurring at this site once, so we can say that there is a possibility of 1 out of 4 for D, that is 0.25, 0.25, and 0.50. So total probability will be equals to 1. So this is how previously, with the probability formula and all the Bayes rule, I explained you how it actually calculates the probability of transitions and emission at each site. Now, using one example of protein sequences, we calculated that how it actually the HMM profile is made, how they calculate the emission probabilities at each site, how they add insertion probabilities and deletion probabilities, and how we can calculate it one by one. So I think it's very simple and clear to learn, but first, to make it more clear for yourself, it's better to watch the part 1 of this lecture before moving into this section. So now these actually probabilities, this is one chart of the HMM profile. So here you can see that the gray cells represent the edges in the HMM diagram, from where we can move from start to M1, and then M1 to M2 or D2, and then so on. These are some values that gray cells can be used from moving from one site, whether that is start, whether that is insertion site, that is match site, deletion site, to the next site. Rest of the clear cells are the forbidden cells. We cannot directly jump, as I showed you previously, that you cannot directly jump from S site to this site, right? So this is not allowed in HMM profile, right? So, and in last, you know that there are a lot of different applications in bioinformatics for predicting of protein profilings, or gene DNA sequence analysis, or horizontal gene transfer, or predicting the conserved sites in a protein sequence, and the repeated CPG islands in a genomic region. So these are different bioinformatics or molecular biology applications where these HMM concepts can be used to find the potential, whether protein family or any potential horizontal gene transfer, or predicting a new conserved site for protein sequences. There are a lot of tools available online which we can or you can use for analyzing your question of interest. For example, here you can say blocks, HMM-ER, HMM-Pro, SciBlast, PFAM. This PFAM is one protein domain predicting site, and so on. So there are a lot of different tools where you can where they use HMM models for predicting or answering their question of interest. I hope this whole lecture was clear to you. Thank you very much. Assalamu alaikum. Bye bye. Thank you.